let me know.
Yeah, I'll let you know in a second. It's just loading.
I'm sure it's the Zoom saw. So.
Okay, it's live. Go for it.
Awesome, thanks. So, hello everyone. I'm Antonio Majdoncic, and I'm a postdoc in Lab at Cold Spring Harbor Laboratory. My work is titled Statistical Correction of Input Gradients for Black Box Models Trained with Categorical Input Features. So, deep learning shows great promise for regulatory genomics. In regulatory genomics, one common task is to train a neural network to take DNA sequences as input and have it predict a regulatory function, such as a transcription factor binding. Unlike previous methods like position weight matrices, which are specifically designed to uncover binding motifs, deep learning methods can learn the same motifs, but also their complex dependencies, such as regulatory grammars or soft motif syntax, using a single flexible model. So neural networks are complicated models for a very large number of parameters. They are basically black box models. A good model should not only have high performance in its prediction task, but also have an ability to indicate how its decision was made. So ideal model should really have two important properties. It should make predictions with high accuracy, and it should also be interpretable. So what are the ways to understand what the model is learning? Well, we can try to understand neuron activity. For example, we can visualize the first layer of a CNN, or we can find what maximizes the activation by optimizing the input. We can use attribution methods. This is the use of gradients or perturbations to capture the sensitivity of predictions with respect to the inputs. We can interrogate the network by testing different hypotheses about the patterns that the network is learning using in silico experiments. So in this work, we focus on the very popular gradient-based attribution methods, which rely on input gradients. So gradients are important. They are used in many attribution methods. Typically, a sequence is encoded to a one-hot representation. After training the model, we can then compute input gradients. Each position having 4 possible nucleotides will have 4 partial derivatives. We can now use these gradients to for different attribution methods, such as saliency, integrated gradients, smooth grad, etc. Now, if we plot pure input gradients, we call it a saliency map, which is a very popular sensitivity measure. It can uncover patterns such as motifs. However, gradient-based attribution maps, they are notoriously noisy. A realistic map often looking something like this at the bottom. So why gradients noisy, and what can be the sources of this noise? Well, first, the function that is learned can be noisy. This is usually addressed by regularization methods such as network weights regularization, Jacobian regularization, adversarial training, randomized smoothing, attribution priors. Then perhaps they could be learning poor representations, like poor noisy features, learning shortcut representations. Attribution methods themselves may have fundamental issues. That's why some of them are trying to satisfy some plausible axioms or any other unknown reasons. So in our work, we identify a new type of noise that we get to understand deeply and we correct for it. So something very interesting happens when feeding data with constraints. This data lives on a lower dimensional manifold within the full parametric space of higher dimensionality. One such data is central in genomics. DNA and RNA sequences, they are encoded as a one-hot representation, and this can be interpreted as the probability of each nucleotide at each position. So the sum of each column must equal to 1. Because of this constraint, the data lies on a lower dimensional simplex given by this equation below, which represents a flat plane. On the right, you can see a toy example that shows geometry for 3D input. All points in this plane represent different combinations of nucleotide probabilities that sum to 1. Now, something very interesting happens when you train a network on this type of data. The function that a network learns will be well defined on the simplex, where the data points live. However, the function has freedom to express any random behavior outside of the simplex because there's no data there to guide the function. So here in the animation, we illustrate that by showing two very different functions with different angles with respect to the simplex. Here lines represent gradient flow. It's basically a way to illustrate the function in space. An important point is that this random function behavior outside of the simplex, it does not affect predictions because all data lie on the simplex, including the test data. But this could introduce issues in the gradients, which do rely on the function behavior outside of the simplex. So our main hypothesis is that this random off-simplex function behavior can introduce a source of noise in input gradients. Our main contribution is a correction of this noise. We suggest removing the random orthogonal gradient components, which are not guided by any data, by simply subtracting the original orthogonal component from the total gradient. So let's derive the correction for the one-hot DNA sequence, which has for a manifold 3D plane in 4D parameter space. We set our DIs to remove the orthogonal gradient component, and this is how it's done in vector algebra. We insert the general expression for gradient g of a function with 4 variables. We also insert n, the normal vector, which is different for every simplex. It is unit vector perpendicular to the simplex, carrying the information about a local direction of the surface. Finally, after some simplifying, we get the result for the correction. Here, for the one-hot DNA example, you basically should take the partial derivative of each nucleotide and subtract them by the mean of the partial derivatives at that position. So this is a theoretical prediction about a hypothesized noise, its nature, and how to correct it. We first test the existence of our hypothesized noise and the corresponding correction for it on synthetic data. Investigating in silico models can be very useful. We have the ground truth and we control the complete information, so we can quantify the efficacy of attribution maps. Our synthetic data set consists of positive and negative label sequences embedded with different sets of motifs. We test 4 different neural network models, one that is more shallow, containing 2 convolution layers, and deeper one with 4 convolution layers. The shallower network is designed to promote learning of whole motifs in the first convolution layer. The deeper network tends to learn more distributed representations. For example, to learn partial motifs and then assemble them in deeper layers. For the first convolutional layer in both networks, we pick from the set of 2 activation functions, either ReLU or exponential. And this gives us 4 different models in total. Now the results. First, we confirm that random angles between the learned function and the simplex are indeed present. Here we plot the distribution of angles between the gradient vector and the simplex at each nucleotide position for one of the networks. You can see that the angles are randomly distributed, but centered around 0. So most angles are actually less than 25 degrees, which largely supports the use of gradient-based attribution methods. But there is a long tail of much larger angles, which we believe is a major source of gradient noise. Now let's finally apply our gradient correction to saliency maps. Here you can see our correction in action. On the left, you can see one example. The top sequence represents a saliency map before the correction. In the second row, we plot the size of angles between the gradient and the simplex. The third row shows the correct saliency map. This map is indeed much more similar to the ground truth, which is in the bottom row. So our gradient correction removes spurious noise and improves motif definition. On the right, you can see another example. So these are just anecdotal examples. To confirm the efficacy of our gradient correction, we perform the full statistical analysis on all sequences under all models. And here are the results for saliency maps using cosine similarity as a metric. Cosine similarity treats attribution maps as vectors, and it compares the similarity of saliency to the ground truth by calculating cosine between the 2 vectors. Cosine value of 1 corresponds to perfect match. 0 is for random guess. Here in the figure, the y-axis represents the improvements, which is the difference in the cosine similarity after correction minus before correction. Every point represents a different run, for different network architecture. You can see that most models lie on the green side, where positive improvements are, which demonstrates that the correction consistently improves the interpretability performance across most models. Our results are robust under different similarity measures, which are left to right, cosine similarity, AUROC in the middle, and AUPR on the right. We obtain similar results for integrated gradients, which is another gradient-based attribution method, here shown in the second row. So our correction definitely brings better attribution maps, so it does seem to be working. But is the mechanism of this noise really as we hypothesized? To confirm that, we need to establish the association between the large noise and large angles. And this figure shows that. Here we plot improvement in the interpretability at the positions where motifs are embedded, and you can see that the improvement really shines for larger angles. This confirms our hypothesis that the gradient noise can arise from the arbitrary function behavior of the simplex. Now we are ready to test how our correction works with in vivo data. In vivo, it's a little bit problematic because we do not know the ground truth. So we resort to using an ensemble average of the saliency maps across many models as a proxy for the ground truth. We are assuming that ensemble averages provide more reliable attribution maps compared to any individual run. We study 10 transcription factor ChIP-seq datasets framed as a binary classification task. A correction indeed improves saliency maps in vivo. Here is an anecdotal qualitative example. The top sequence represents the saliency map before the correction. The second row shows the corrected saliency map. This map is indeed much more similar to the ground truth proxy, which is at the bottom. We evaluate this improvement across all nucleotides and all sequences in the following way to get a full statistics. So we calculate difference vectors between the saliency scores and the ground truth proxy at each nucleotide position. To get a single number representing how different they are, we take the L2 norm. Figure on the right shows average values of such differences as a function of angle. The results are shown for one selected protein, E2F1. Here, the red line represents the difference between the uncorrected saliency maps and the ground truth proxy. Green line is the same for corrected saliency maps. We see it has a reduced difference, especially for larger angles, bringing it closer to the ground truth. So we investigate a lot of different other properties in experiments, and we cannot go over all results in the paper, obviously, but I'm going to show you one more interesting experiment. So by increasing the weight initialization standard deviation, we notice that it results in a larger average angles after the model is trained. This is the blue line in the figure. The classification performance here in red stays roughly the same, albeit with a tiny decrease as initialization values increase. For this experiment, we hypothesize that a larger random initialization leads to larger initial orthogonal components, which never get corrected afterwards because there's no data of the simplex to guide the function. In conclusion, we identified a new type of gradient noise present in data with constraints that arises from a random orthogonal component, which is a common case in biology, including DNA, RNA, and proteins, where one-hot representation is used, as well as in other fields such as NLP with one-hot representations. We derive the correction for flat probability simplex, but if any manifold, including curved ones, that constrains the data can be determined, then a similar gradient correction can be derived. Finally, I want to stress that this method only corrects gradient noise that arises from arbitrary function behavior off of the data manifold. It does not correct when a model learns poor representations. Yeah. So finally, basically, if you're using saliency maps, integrated gradients, or any other gradient-based method, you should definitely use this correction. It's very simple to implement, like one line of code, and it has a theoretical foundation, really. Finally, I'd like to thank members of CoolLab, led by Peter Koo, and also Justin Kinney, David McCandlish, and Anapos Fai for helpful discussions. If you're interested in postdoc opportunity in CoolLab or Kinney Lab, you can see emails below.
Great, very interesting, Antonio. Can you say a bit about whether this has implications for some of the other approaches for doing CNN interpretation? So in silico mutagenesis or deep lift or deep sharp?
Right. So, I mean, mutagenesis would be just like, replacing, sort of like swapping the nucleotide. So this is really for like gradient based methods. So basically any method that is using input gradients this is definitely applicable there. The output doesn't matter. It could be categorical. It can be regression. We tried an RNA regression, for example, task with and it worked there. So in all matters that it uses gradients, input gradients. Yeah. That's the formulation of our theory.
Cool. And something else I was wondering while you were going through this was whether if you took the input and as kind of a weird first layer, you did a softmax across channels. So you're basically saying, oh, sorry, not a softmax, you normalize. So that would mean that you enforce that even if you had an input that did not sum to 1, you would force it to sum to 1. Would that do something like equivalent to this? Where
you mean like you're talking about, for example, by trying to. So I mean, this is really about input gradients, right? We did try, for example, regularization during the training. That's maybe something similar, and it works, right? The problem with that was that in regularization you have some parameters that you need to tune. You can easily over-regularize, like in any regularization, right? If you over-regularize, your cost function will only care about that thing, not like classification task or regression task. So, right. But this is very important to say that this is. We think the most efficient implementation is basically post-training. So you need to notice that here we did not touch the model that is trained. You can literally take a model that somebody else has trained, okay, use the weights and everything, and make produce some attribution maps that are gradient based, and you can apply this correction. You don't need to train a network. Right? We did try, like I say, we used, for example, angles. We just added some absolute value of angles as a penalty to cost function, and it does a similar effect. But we think it's more complicated than just simply let it train and then doing this correction in one single line.
Gotcha. Great. Yeah, that's very useful. Thank you. Okay. If there are questions, feel free to put them in the Q&A, and maybe Antonio can answer them.
Yeah, of course.
Next. So next up, we have Zemming Lin from Facebook AI. So he's going to be talking about deep generative models of protein structure create new and diverse proteins.
Hi. I can't seem to share the screen.
Yeah, I think, Antonio, you may need to stop sharing.
Okay.
I just stopped.
Yeah, great. Thank you. Yes, perfect.
Can you see my mouse as well?
Yes, I can see your
mouse. Okay, great. Hi, my name is Zemming Lin. I'm actually a PhD student at NYU under the supervision of Yang Li Kun right now, and this project was done partly through an internship with Facebook AI research with my colleagues Tom and Alex, and it's on deep generative models of protein structures create new and diverse proteins. One of the challenges of protein design is how we can propose interesting and valid protein backbone structures. And so this work is a way to tackle that. We ask ourselves the question, can we use neural network models to generate realistic protein structures? And if we can generate these proteins, how do we evaluate the generations? Because we can't just go printing every single generation that we make. And it's especially hard for neural networks to do this because it's hard for the models to learn these complex three-dimensional interactions to actually generate proper sort of compact proteins with nice secondary structure. So here we actually show some of the structures that we ended up generating, and for the prior work, you can kind of look at how these compare to the prior work, and some of the sort of most interesting papers in this landscape include Anand et al. In 2018, who uses a GAN, and I think this is one of the first works using neural networks to generate protein structures, and you can see that it's not quite compact. You don't get good secondary structure, and a lot of the proteins are really missing even tertiary structure. More recently, Iguchi et al. Uses a coordinate VAE. So this is a VAE type model where you push the input, which is a histogram, into a bottleneck, which kind of gets spread out into a Gaussian ball, and that reconstructs directly the coordinates of immunoglobulins. And the downside of this is, even though you're able to generate realistic protein structures, they only come out of a singular fold. And more recently, I think TR design is one of the more interesting works in this area, which inverts the popular structure prediction model, TR Rosetta, to generate very realistic looking structures. And they do this by sort of maximizing the KL divergence between a background distribution and the generated histograms. And they do this like multiple rounds, kind of using a Monte Carlo method. And this generates fairly realistic proteins, although they show that most of them are similar to structures in nature. And as a summary of our contributions, we kind of provide a different way of generating coherent and diverse protein structures using variational autoencoders. And then we found something really interesting with this model, which is that after we design these structures, we can, or after we generate the structures, we can design sequences for them. So we're only generating the backbones. We ask Rosetta Fast Design to generate some sequences, and then we validate this with AlphaFold 2 as an oracle. So the AlphaFold sees only the sequences, and it predicts actually with high confidence that a lot of the structures are similar to our generated structures. And finally, we look at the sequence identity of the sequences that we generated, and they're actually quite low with natural proteins, which we found to be pretty interesting. So to tell you guys a little bit about how this model works, the data that we use are SCOP domains filtered to length 128 examples. And this is sampled evenly at the family level to remove some of the bias with protein structures, since we have a lot of structures for a very small number of proteins that are quite similar. The features that we use are histograms and interresidual and torsion angles. And so the distance distribution, the backbone torsion angles are introduced in AlphaFold 1, and the interresidual orientations by TR Rosetta a bit later. The models that we use, first, we have as a baseline, the traditional variational autoencoder. And this architecture takes in your X as a histogram. And so these are all 2D maps, and then we can use a convolutional encoder and a convolutional decoder to generate a histogram output. The more interesting sort of newer VAE models include this very hierarchical VAE. And the way that these VAEs work is that they have multiple levels of latent variables. So you can see down here, maybe you get input and X, and this X goes through a ResNet at each level in the ResNet. So maybe the first level is a 16 by 16 quote image. The second level is 8 by 8. The third level is 4 by 4, etc. It produces a posterior and a prior. And the way that these work is that so for example, the 4 by 4 level, it'll come up here and then generate a latent variable up here and pass it along. The posterior takes the information from the more coarse-grained latent variables in the higher levels, and also the features from your input. So the posterior is actually a Z that's dependent on the Z's of the prior, more coarse resolution latent variables and your input features. And the prior is only dependent on the Z's that are sampled in the more coarse resolution latent variable, the generator stack, if you will. And there's a last loss that pushes the prior and the posterior together. So it tries to minimize the KL divergence between them. And during generation time, you can simply sample the Z's, you predict a prior, sample another Z from the prior at the next level, and so on and so forth. And the model that we kind of found to be more effective is the VQVE architecture. It's possibly hierarchical in the same way. You can generate an 8x8 sort of feature latent variable map, and these are quantized and then sent through the decoder. There's a sort of heuristic loss that takes your quantized vectors and forces them to be separated from each other and also produce good-looking reconstructions. And then from these quantized latent variables, you can learn a prior. And so you can think of the quantized latent variables as sort of a sentence, because you have 8x8 of them, so 64, and each one has maybe 512 tokens, and then you can just learn an autoregressive transformer language model to model your prior. And so here we learn exactly that. And during generation time, we can greedily sample from this language model. I think it's a 6-layer transformer. We just took the typical parameters from the attention is all you need paper, and then you can take the tokens that we generate and decode it into a protein. And this protein is made up of an energy function, which is the distogram, sort of the constraints that we generate, and also the RFX, which is a term for the chemical validity of the structure. And this is, a very common thing that's used in the Rosetta structure modeling community. RFX is generally just the default Rosetta Ref 2015 score, and we can minimize the coordinates of the structure to satisfy this energy function. We did run into one problem, which is that the hierarchical VAs and VQVAs tended to overfit. You can give it as many latent variables as it wants, and if you give it enough latent variables, it's just going to memorize your inputs completely, and so the validation loss we get is going to be 0, but it's not going to be able to do good generations because the generator is only recapitulating whatever's in your latent variables. So therefore, we had to find a different way to check the goodness of our models. We just fold it using Rosetta and look at a few metrics like the Rosetta energy, long-term contacts, long-range contacts, and secondary structure. And the way we do this is basically exactly like TR Rosetta. We generate the angles and the histogram. We smooth the restraints out and add them as restraints to a minimization function, and that minimizes the coordinates with respect to these energy functions. We get a few different models, and then we fast relax the best one goes at a energy function. And the backbone only models, so you can either use the interresidual orientations, or you could just use the backbone dihedral angles as constraints. We found that these backbone dihedral angles actually performed a little better, but only if you really smooth it out by just fitting a single von Mises distribution to the angles. So you're just making it kind of a unimodal distribution. And so we can look at the quality of our model compared to the hierarchical VAE and the VQVAE compared to a few baselines. The first column is RFX less than 0. So this is an energy output by Rosetta. If it's negative, it's good. You actually see that HVA does a little better in this. It does the best, and the average energy is quite low. But then on sort of visual analysis, we found that the percent of alpha helices that the HVAE generated were quite high. So it's generating all these alpha helices that were very energetically stable because they don't clash with each other, but they're also not very interesting. And so after iterating on this, we found that the VQVAE models were able to generate proteins with these MR-LR contacts. So those are medium or longer range contacts. I think kind of a 12 residual cutoff or polar bonds. It's kind of a a way to measure how compact your protein is. If it's contacting everywhere, you're going to get a higher number here. And so they were generating much more compact proteins, and it was really the only model to be able to generate a lot of beta sheets. You can see here that the proteins on average, have a much different beta sheet distribution compared to the baselines. Here are some sort of cherry-picked generations. We picked the best ones, and even out of the best ones, the HVAE models were really only able to generate alpha helices, where you see a bunch of beta sheets for the VQ. Is there a question? Okay, no. And we also looked at diversity. So we can separate them into this mainly alpha, mainly beta, or alpha beta proteins. And you can see that most proteins pairwise diversity, according to TM score, that's structural alignment, is quite low. But for the beta proteins, we found some evidence of overfitting. We suspect that this is probably due to our data set, and we're looking into ways to mitigate this. And on the right side, you can see the TM score with the nearest neighbor in PDB, and most of them are not 1, which means that we're not really recapitulating the training set, although we do have a lot of proteins that are quite close, and none of the proteins are really of a different fold. And so these are folds that exist in nature, but not the exact proteins. Finally, we decided to do this design experiment. So fast design is quite a slow procedure because you have to design a lot of sequences and then pick the best ones. And so we did this on just 38 structures that pass a heuristic quality filter from the VQVAE backbone model. And we found that if you give these designs to AlphaFold, you see that AlphaFold can predict high confidence structure matches for 9 of these designs. For the ones that had low confidence, I don't know how much we can say about them. So we decided just to look at these 9, and 8 of the 9 had pretty low RMSD with our generations. And so the first column, you can see the generated histogram, and this is provided to Rosetta as a constraint. In the second column, you see the sequence from fast design after it was run through AlphaFold, the histogram, and they look quite similar. The third column, you can see the AlphaFold predicted protein overlaid with our proteins. The fourth column is the nearest neighbor in PDB overlaid with our proteins. And the fifth column, here I circled in red the number of hits against UNIREF 50 and Magnify when we ran these sequences against these 2 databases with Jackhammer. And you can see that we found really just 1 sequence for this protein and no sequences for this one. And even though they had a match here, there's very low sequence identity. So we wondered if this was a quirk of fast design, or if this was a real effect that our models were generating proteins with sequences that had low identity with metagenomics databases. And so we ran some natural proteins through them. And if you look at the natural proteins, I think half of them tended to get 0 matches, which kind of, I think it probably means that the lowest energy sequence was not the correct sequence for the structure. But for the ones that didn't have, or that did have some matches, our designed proteins had very few. Most of them had under 10 matches, and the maximum was 100, whereas the natural sequences had at least 100, up to tens of thousands of matches. And even when there were matches, the sequence identity was quite low. So this gives us some hope that our model might be able to generalize a little bit outside of existing sequence space, and it's able to generate structures that are somewhat novel. Yeah, so in summary, we found that the VQVAE models can generate realistic proteins. We introduced a few metrics like Rosetta energy, long-range contacts, and secondary structure to validate the generated proteins. And we found that sequence designs agreed with the generated structures, and there was low sequence identity with natural proteins.
Very cool. So to the audience, please feel free to put questions in the Q&A. I'll start with one while people are thinking of theirs. So are you or others thinking about trying to do this sort of jointly with sequence? So you can imagine trying to generate, have a joint generative model over structure and sequence. I don't know if that's too hard to do, but
I think my hope is to actually take this work in the realm of being able to do conditioned structure generation. So my belief is that structure is sort of the underlying sort of thing in biology. If you want to design a drug, you want to create a structure that binds to another structure. So if you can do good conditioned structure generation, then you can put that together with perhaps like a sequence design method and do things and that way. So it's not together with generating sequences, but it would be to generate a structure of your choice first and then to generate sequence for that structure.
Right. So Yongjin Park asks, did you find anything interesting in the latent representation?
We didn't really do a lot of analysis in the latent representation. We looked at it a little bit. There wasn't anything obvious that seemed to have popped up.
And then there's a question in the chat from Pinar Demetsi asking, my understanding is that what makes VQ-VAE distinct is that it learns discrete latent representations. Right. Is there a biological intuition for why that seems to help with structure generation?
My intuition is that it's maybe learning sort of the underlying grammar for the generated structures. So it's maybe able to learn, like the structural motifs that make up a protein. And then if you're able to generate a sequence of these motifs, you're able to generate a good structure. But yeah, further work is needed to see if this is actually the case. It's kind of hard to analyze the VQ models because they're no longer continuous. So it's hard to do the latent variable interpolation, for example, and the prior is this autoregressive language model. And so it's very difficult to analyze these latent variables.
All right. And my final question is going to be, so right now you're doing, I think you said 128 amino acids, is that right? Like, are you thinking about doing bigger or variable lengths as well?
These are already variable lengths. So you can actually, what we ended up doing is you can predict the where the sequence ends, kind of like where mask tokens are, and then your model actually just automatically generates variable length protein structures. And we tried it on some longer structures, but I don't think we were able to get it to fit well enough. My suspect is that the inductive bias of the histogram is actually not very good. So the model won't be able to learn the three-dimensional constraints as well. And we're trying some of the AlphaFold style techniques to see if they have a better inductive bias.
Great. Okay. I think that's a good time to move on. Thank you again for the talk. So next up, we have Justin Keeney from Cold Spring Harbor Labs, who will be talking about MAVE-NN, if I'm saying that right, learning genotype-phenotype maps from multiplex assays of a variant effect. So we can see your slides, Justin, all yours. Can't hear you currently, however. Not hearing you yet.
Can you hear me now?
Yes, there we go. Great.
Okay. And does the
slides were looking good just now?
Slides look good? Okay. Can you?
Oh, well, no, we lost it again. You had it just now, but I think you need to screen share again.
Yeah. Okay, can you see it?
Perfect. Yes, and I don't see your mouse currently, but maybe you can.
Mouse. Okay.
Oh yeah, now I see it. Now I see it. Great.
Now you
Perfect.
see it. Okay.
Yes. Great. Thanks.
Okay. So. Okay. Well, I'd like to thank David and the other organizers for giving me this chance to talk to you today and for organizing this conference more generally. I really look forward to this conference being back in person sometime in the near future. I kind of miss that energy, but this has been great so far. So I am an associate professor in the Simon Center for Quantitative Biology at Cold Spring Harbor Laboratory. Today, I'm going to talk to you about the work that was spearheaded by my recently former postdoc, Amar Tareen, and is now being taken over by my current postdoc, Mahdi Kushkabagi. And just to do a little advertising first, I want to let you guys know that there's a new job opportunity in the Simon Center So Peter Koo and I, Peter Koo's a colleague of mine just down the hall. We're looking to jointly hire a postdoctoral fellow in deep learning, biophysics, and genomics. So the goal of this position is going to be to try to bridge the kind of fundamental conceptual divide between deep neural network models in genomics and quantitatively interpretable biophysical models of gene regulation. We have a job advert out in Nature Careers and the job archive. If you're interested, send us an email. Okay, so one of the primary focuses of my lab is trying to understand how to quantitatively model data produced by multiplex assays of variant effects, or MAVs. So MAVs encompass a wide variety of experimental methods for studying sequence-function relationships. These include deep mutational scanning experiments for studying protein sequence-function relationships or for studying structural RNAs. They include massively parallel reporter assays for studying transcriptional regulatory sequences, as well as regulatory elements in mRNA that regulate post-transcriptional processing. And then there's also a wide variety of other bespoke MPRAs, such as for studying initiation of DNA replication and bunches of other things, other bespoke MAVs. So despite the vast diversity in both the set of sequences that MAVs are used to analyze, MAVs are used to study, as well as the set of kind of experimental outputs, they do share something in common, which is that almost all of these methods produce large but noisy and actually very highly nonlinear data sets. So there's a common challenge in analyzing MAVE data, which is there's a basic need to deconvolve the underlying sequence function relationship that you're interested in from the very strong experimental nonlinearities and noise that can result from these sorts of experimental methods. So to address this need, my group has developed a Python package called MAVE-NN for inferring quantitative models of genotype-phenotype maps, or equivalently sequence function relationships, from MAVE data. So we've put a fair amount of effort into documenting the software, which you can find on Read the Docs. Software is open source, available on GitHub, and it can be readily installed from PyPI by just doing pip install Maven. We'd love to hear any feedback any of you guys have if you have a desire to use it. If you want to read more about the method, please see our recent bioRxiv preprint by Amar and company. So Maven implements in order to do this, Maven implements a conceptually unified approach for modeling genotype-phenotype maps from all types of MAVE experiments. So this type of modeling approach, we call it a latent phenotype model. So latent phenotype models have the following structure. They assume that each sequence X is mapped to a low-dimensional latent phenotype variable phi via a deterministic genotype-phenotype map. After that, the latent phenotype then determines the experimental measurements through a stochastic readout process. So the measurement is a stochastic, often very highly nonlinear readout of the underlying latent phenotype. So you can think of the first part of this, the genotype-phenotype map, as carrying out a very large amount of dimensionality reduction. If you think of sequences as very high dimensional objects, the latent phenotype is a very low dimensional object. The genotype-phenotype map is compressing all this information in a very high dimensional space down to a very low dimensional latent phenotype. Then the measurement process is the thing that couples the what you actually measure in your experiment to this underlying low dimensional latent phenotype. So these models are implemented and trained as neural networks using a user-friendly API that interfaces with a TensorFlow 2 backend. So Maven, now this structure, this model, the latent phenotype model structure supports a very wide diversity of genotype-phenotype maps and measurement processes. So currently Maven supports models with linear models based on sequence features. These include additive features, nearest neighbor features, as well as pairwise features. Maven supports black box neural networks, such as deep multi-layer perceptrons, and also supports custom genotype-phenotype maps that can be defined through subclassing. And these can represent, for example, biophysical models of protein folding and binding, or biophysical models of gene regulation, of transcriptional regulation by promoters or enhancers. Maven currently supports two different types of measurement processes in order to support both continuous and discrete experimental measurements. So global epistasis regression is modeled, is used to model MAVE data that contains continuous measurements. So the global epistasis measurement process assumes that the measurement is a noisy readout of a nonlinear transformation of the underlying latent phenotype, whereas the measurement process agnostic regression approach is used to analyze MAVE data that contains discrete measurements. So here, the probability of each of a certain discrete number of measurements is represented using a very flexible function of the latent phenotype. And I want to emphasize that when Maven fits models to data, it infers both the GP map and the measurement process simultaneously, and this is quite simple using TensorFlow. Finally, on the conceptual side of things, Maven uses an information theoretic framework for quantifying model performance across all MAVE datasets. I want to emphasize the importance of this. The diversity of MAVE datasets and their noisiness means that you cannot use standard measures. You cannot use standard metrics that are commonly used in machine learning, such as R-squared, mean squared loss, or accuracy. If you want to support all the types of data sets that, all the types of data formats that MAVE spit out. This information theoretic framework does handle all types of possible MAVE data sets. So I'm going to describe how this framework works. So intrinsic information is defined as the mutual information between sequence it between measurements and the sequences themselves. So this is intrinsic to the data set. It doesn't depend on the inferred model. Predictive information quantifies the performance of the genotype-phenotype map independent of the measurement process. It quantifies the mutual information between the MAVE measurement Y and the value of the latent phenotype predicted by the genotype-phenotype map. And then finally, there's a quantity that we call variational information. This is actually just an affine transformation of log likelihood, but it's transformed in such a way that it can be directly compared to the other information theoretic quantities. And in fact, in the large N limit, you can show that this model performance inequality holds. That is, the intrinsic information is always greater or equal to the predictive information, which is always greater or equal to the variational information. Moreover, equality obtains between the intrinsic information and the predictive information only when the genotype-phenotype, the inferred genotype-phenotype map is correct. And equality obtains between the predictive information and the variational information only when the inferred measurement process is correct. So again, if you're interested in seeing more about this, please refer to our reference. Okay, so now I'm going to quickly go over 3 applications of Maven. The first is application to this famous deep mutational scanning experiment of protein GB1 performed by Olsen et al. And Ren Sun's lab back in 2014. This is one of the best quality deep mutational scanning data sets out there. So they used in vitro transcription, translation, and mRNA pull down in RNA display, and then affinity pull down to assign log enrichment values to about a little over 500,000 coding variants of the protein GB1. So by fitting a model to these data using Maven, we can get an additive GP map that describes the mapping between GB1 sequence and the latent phenotype. We also get a measurement process for this experiment. And as you can see, the log enrichment is a highly nonlinear function of this inferred additive GP map. And moreover, the noise in the experiment is very heteroscedastic, and it's also asymmetric. But Maven's GE measurement process is flexible enough to handle that. This model fits very quickly. So on a data set of 500,000 sequences, it takes under 10 minutes. For about 100,000 sequences, it takes a minute, and so on. And it's very accurate in the sense that it can very precisely recover known underlying parameters, and you can test this for any inferred model because all latent phenotype models inferred by Maven can be used as dataset simulators that you can then repeat the inference process on. The second example that I'm going to talk about is this RNA-seq-based massively parallel reporter assay of 5' splice sites in human cells. So this is a collaboration between my lab and Adrian Krainer's lab. We published a few years ago. So here we use a mini-gene reporter assay to measure the percent spliced in values from essentially all 33,000 possible 5' splice sites. And we did this in human cells. So for each 5' splice site sequence, we got a percent spliced in value. It's continuous data. So we did a GE regression, but now this time with a pairwise genotype-phenotype map. There's an additive component to this map, as well as a component that describes interactions between all possible pairs of positions within this non-nucleotide sequence. And I'm showing the inferred model here. This is the inferred GP measurement process. And what we find is that as you increase the flexibility of the model, you go from additive to neighbor to pairwise to black box, the model performance does increase. It's not surprising, but it highlights the importance of having a single framework in which you can fit a wide variety of genotype-phenotype maps without mucking with everything else. And Maven allows users to do this in a very straightforward manner. Finally, I want to highlight a third application, which is the application to this very early MPRA. So this is a fluorescence activated sort-seq based MPRA that I performed back in 20 and published back in 2010. What we did is we took this 75 base pair region of the E. coli lac promoter, and we created a library in which this was mutagenized by about 12% per nucleotide. It was used to drive GFP expression, put that into cells, sorted cells based on their fluorescence, and then sequenced the mutated, sequenced the variant promoter sequences within each of 10 bins. So the resulting data set for this actually looks quite different than the others. So here, each variant is associated with a vector of counts across the 10 possible bins. So to analyze these data, we use measurement process agnostic regression, MPA regression, with a custom GP map representing a biophysical model. This is a quick summary of the biophysical model. And by fitting, using MAVEN, we can infer the energy matrix models for CRP, energy matrix descriptions of the sequence specificities of both CRP and RNA polymerase, which is shown here. I want to emphasize, these aren't just statistical motifs. These are energy matrices with whose parameters have meaningful units of kcal per mole. We also infer a specific protein-protein, in vivo protein-protein interaction between CRP and RNA polymerase. And this is the inferred MPA measurement process. As you can see, it's kind of a weird mapping between the latent phenotype and these 10 bins, but MAVEN is able to learn that at the same time that it learns the biophysical model. So with that, I'd like to thank the members of my lab, particularly Amar, who spearheaded this work, and then Madi, who's starting to take it over, and then Dave McCandlish, who's a fellow colleague of mine here at Cold Spring Harbor, who's also a collaborator on the paper. Thank you very much.
Great, thank you, Justin. It's really cool work. I was going to ask about for the information theoretic quantities that so that mostly comes down to mutual information. Are those being estimated under a parametric model, or you have some non-parametric way of doing that?
That's a very good question. So you can think of variational information as to the extent that the inferred latent phenotype model is parametric, the variational information is the variational information is it is like the model's parametric estimation of mutual information. Predictive information, we actually estimate that using non-parametric methods. So using this kth nearest neighbor method that was published by Kraskoff et al. Back in 2004. And now estimating intrinsic information is kind of a difficult task. MAVEN does not implement specific estimators for intrinsic information. If you want to estimate that, you kind of have to do it on your own because it's going to be different between each different data set. But if you do it, it provides kind of the maximum level of performance that any genotype-phenotype map can have on the data.
Right.
Yeah.
So that seems like it would be very useful for assessing, parametric models that you're actually using.
Well, in particular, I want to emphasize that if you're like, you can't use things like accuracy because, or just R squared, because like there's some ceiling and you need to know what that ceiling is. And essentially intrinsic information provides that ceiling. Yeah,
I agree. We miss this a lot in machine learning. So the other thing I was going to ask about was these latent phenotype to measurement maps that you're inferring look great. I was wondering, are there constraints you're putting on that? Is there something about like monotonicity or something like that that's helping the model sort of get to those nice fits?
We do have an optional monotonicity for the constraint for the GE measurement process, but honestly, it really ends up kind of mostly determining a sign.
Okay.
more generally, we're now working on building up a compendium of measurement processes that actually represent different kinds of experiments. And these are kind of the two basic ones, but there's a lot more that you can imagine.
Great. Okay, awesome. So we're at time. So we have a 20-minute break. We will resume in 20 minutes, which is 2:20 p.m. PST, I believe, but maybe a different time in whatever time zone you are in. So we'll look forward to seeing you all back in 20 minutes. Thanks again to the speakers from this session.
So there were a few people that asked if they can check out their setup and make sure that they can share screen. If you want to do that now, we have time to do that.
That would be great. Can I try sharing my screen?
Yeah, perfect. We can see it.
Okay, thanks.
Hi, could I also test my slides?
Sure.
Okay, so my audio is working.
Yeah, we can see it. Perfect.
Sorry, did everyone see my slides there for a moment?
Yeah, looks great.
When it was in full screen as well?
Uh-huh.
Okay, great. All right, see you after the break.
See you.
I'm sure it was great to see you at the Stanford.
Oh yeah, likewise.
Now my vacation is over. I became really busy, going over hundreds of emails.
Yeah. At least, yeah. I think the weather was nice when you visited, right?
Oh yeah. People said that there were some, rains and
APIs because they like checking their emails as opposed to doing the implementation work.
Yeah, hopefully next year we'll be able to do this in person. Probably a hybrid meeting. I think there are some advantages of being able to screen it for free. We get a potentially more international audience. But
are you talking to me? Sorry, Aisha, are you talking to me?
I'm just commenting generally.
Oh, I see.
Since we
Yeah, I totally agree. I think if we can keep on streaming, it would be great in some sort of hybrid.
Yeah.
Hybrid is the hardest.
Mm-hmm.
Yeah, it needs to be either one way or the other. Okay, I need to go to a meeting now. Okay, see you later.
I like the idea of having it be focused on being in person, but then also providing some sort of live stream in a limited format.
Yeah, yeah. I think it's feasible. I think we did it once a few years ago. I think that was it the first Mlcb or the second one? I can't remember.
Yeah, I think 2019 is it's only been in person once since it like started to be independent, and I think the first one we also streamed.
Yeah.
Yeah.
Yeah, now there are even better devices and stuff to enable that. So I think it's doable, actually.
Yeah, back then I think David found some, it was like some hack that he found to record and then like gonna upload at the same time or something like that. I don't think it was Zoom. I don't think there was Zoom back then.
Yeah, it was some open source.
Yeah.
Yeah. But it's been good. It's been pretty consistent attendance. It's been in the hundreds, 200s. I like how 1,000 people sign up, but then most of them don't end up making it.
Yeah, I wonder what happens in between.
Well,
I
you
think
don't know
it's
that. All you know is the maximum number of people who are attending at a time, not whether or not they're slowly mixing in, staying for a few minutes, being like, oh no, and then leaving.
Oh, that's true. Yeah.
At some point I checked Zoom and YouTube, and there was like 100 or 200 people on YouTube as well. So maybe some of them just go through YouTube directly.
Yeah. Yeah. In the future, what we can do is actually have them register directly, like Zoom. A Zoom webinar allows like provides a registration link. So if you do it through that, then as you register, you just get an email to join.
Yeah.
So that way, even if you register late, like it just automatically sends them an email. Because I think what happened was some people signed up today morning and stuff, and like obviously we weren't able to send them links.
Yeah.
Oh, can someone share the YouTube link? It is on.
It's on the website. Nothing changed since the last time, Anshul, right?
Yeah.
So I think the latest one is on website.
Yeah. Every time I stop the live stream, if I restart it, YouTube gives me a new link. So that's the only annoying thing. But yeah, we just have to keep updating it on the website. But so far, we haven't turned it off since Regina's talk. So I think we're fine. Tomorrow's link will again be different. Yeah. The YouTube link, not the Zoom. The Zoom link will stay the same. Great talks, though. Really nice. Good, nice variety of topics and academia, industry, good stuff. So for the speakers of this session, I'm going to set this blue sky timer. You can see it, right, as another video. It will make a gentle sound when you reach 18 minutes. It'll sound something like this. So let's see. Did you hear that?
Yeah, it was not very gentle.
Okay. Yeah, there are much more aggressive ones, but it'll just do, yeah, it'll just do a ping. And then at 30 seconds, there'll be another one. Is that fine?
Sounds good.
That was great.
All right. It's 2:20, so we will get started with the next session. Let me just resume recording. Hold on. All right, so welcome to the last session for today. 3 other great talks coming up. So the first talk is by Phil Franken. It's on graph neural networks to predict molecular carcinogenicity prediction.
All right. Hi, everyone. So my name is Phil, and I'm a graduate student co-supervised by Drs. Brendan Fry and Bo Wang at the University of Toronto and the Vector Institute. And as mentioned, I'm going to be presenting today Concerto, a graph neural network approach to molecular carcinogenicity prediction. So this work was done with myself and my co-authors, Adamo and Lazar, and supervised by Drs. Brendan Fry, Leo J. Lee, and Dr. Bo Wang at the Tsai and Wang labs at the University of Toronto and Vector Institute. So first, I'm going to introduce carcinogenicity as a concept and some properties of carcinogenicity prediction as a problem as a whole. So you can sort of think about carcinogenic compound discovery in 2 modes. So there's a high effect size mode and the high exposure. So on the left, you see this plant commonly referred to as birthworts, and it contains aristolochic acid. And in the early 2000s, it was discovered after it was used as a weight loss treatment that this group of people that were exposed to this treatment would develop a rare form of cancer. And after being tested, aristolochic acid proved to be extremely carcinogenic. And actually, it proved to be one of the most carcinogenic compounds, one of the most potent compounds in the carcinogenicity database. And here on the right, you see the high exposure example where you see a chimney sweeper, an English chimney sweeper. And in 1775, Percival Pott identified that this population of people who are exposed to soot and ash develop high rates of scrotal cancer. And only 2 centuries later, did we learn that it's caused by benzopyrene. So there are 2 main methods of measuring carcinogenicity experimentally. And the first is carcinogenic experiments, which is conducted in animals and usually rodents. And at a high level, animals get divided into 2 groups, and then the case group gets exposed to compounds of interest, and then there's measurements between the number of tumors developed in the case group versus control group. And then this approach is able to evaluate multiple modes of carcinogenicity, genotoxic and non-genotoxic. So genotoxicity just refers to whether this compound mutates the DNA. So your carcinogenicity can be caused by non-DNA mutation methods, and actually alcohol is thought to be non-genotoxic but carcinogenic. And then, so sort of a downside of these experiments is that they're sort of expensive and slow. And on the other hand, we have mutagenic experiments where we have a bacterial culture and there's usually key gene knocked out, and with point mutation or another method, and then the bacterial population is split into 2 groups, and a compound of interest is added to one of the plates, and then essentially the readout phenotype is whether there's bacterial growth, and if growth is observed, then you can sort of infer that there is a reversion of this knockout mutation, indicating genotoxic effects. And the upside is that these experiments are relatively cheap and abundant, but they have lower rates of replication. So a key sort of problem in this carcinogenicity experiment space is the maximum tolerable dose design problem. And the idea is that animals are treated with a maximum tolerable dose of the compound without compromising survival, which often leads to inflated dosage. And if we were to group our compounds into two bins, carcinogenic and non-carcinogenic, it results with compounds with orders of magnitude differences in active dose being grouped into the same bin. And that leads to a loss of information. So instead, we can use this TD50 metric for compound carcinogenicity, which measures compound dosage as a proportion of the animal body weight. And then at its core, carcinogenicity prediction is a graph level regression problem. So given a molecular graph, you want to be able to indicate how carcinogenic is it, meaning at what dose will the animal start developing tumors. So for carcinogenic experiments, every data point requires tens of thousands of dollars to generate, and thus our training set is only 1,000 molecules. So our motivation for this project is that public health can benefit from a low-cost carcinogenicity screening approach, and that can include detecting molecules that are moderately carcinogenic and have low exposure, but still affect the general population. It can be, with that, you can have more informative design iterations in drug development. And finally, you can decrease the number of FDA drug retractions due to carcinogenicity from 13%, which it's currently at. And the way that we can do this is in silico carcinogenicity prediction can prioritize compounds for experimental assessment instead of just testing everything. So now I'm going to talk about our contributions. And it's across 3 different areas. So in terms of data, we reinterpret carcinogenicity prediction as a regression problem instead of classification problem. And this also results in more informative labels. For modeling, as far as we know, we're the first to apply graph neural net approaches to carcinogenicity prediction. We use transfer learning from a large self-supervised model, and then we pre-train on related but lower quality mutagenicity experiments that I mentioned earlier. And finally, for model interpretability, we generate counterfactual molecules using XMOL for model interpretation, and I'll go into more depth what counterfactual molecules are later in the presentation. So in terms of data sets, we use 3 data sets. The first is CPDB, which I mentioned earlier as having only 1,000 molecules. And in CPDB, the authors computed this TD50 metric that I alluded to that measures dosage as a proportion of the animal body weight. Then we use CCRIS for our external validation, which contains 200 non-overlapping carcinogenic compounds to CPDB. And unfortunately, that only has binary labels. And then for mutagenicity data set that we use for pre-training, we use Hansen 2009 benchmark data set, and it contains more than 6,000 molecules, which is 5 times as large as our carcinogenicity data sets. So given a molecule, there's a variety of ways that we can represent it. So the first is sort of a more natural graph representation of the molecule. And this is the same molecule represented as a string, it's called a SMILE string. And then here on the right is this fingerprint binary vector, where every single position represents a substructure, and substructures that are present in the molecule are indicated as such, and others, for example, that aren't are 0. So essentially, it's an alternative way to represent a molecule with hand-engineered features. So now this is a high sort of level overview of our model. We call it Concerto. So it has 3 main parts. The first of which is a Grover. It's a graph neural net transformer. The second is an MLP multi-layer perceptron fingerprint predictor. And then finally, we train this multi-round mutagenicity pre-training objective. So Grover, it's a 100 million parameter model trained on 10 million molecules by Wrong et al., so it's not us. in 2020, and it uses self-supervised learning objectives on molecular graphs, and it uses a graph neural net output and feeds it into a transformer, as described earlier. So the second part is our multi-layer perceptron fingerprint predictor. So it's trained on a concatenation of the Grover representation and then the fingerprint representation. And as I mentioned, fingerprint representations can be thought of as sort of domain expert design features representative of important substructures in the molecule. And then finally, we train this representation using multi-round mutagenicity pre-training. And as I mentioned in the introduction, these lower quality mutagenicity experiments are more abundant, and we use them to pre-train the model. And we actually find that multiple rounds of mutagenicity, carcinogenicity training helps achieve a higher performing model. So I'm going to talk about our results. So first, we evaluated our model on the carcinogenicity potency database test set, and we have two baseline models. The first of which is a fingerprint baseline. So just the second part of our model, the fingerprint, as well as a multi-layer perceptron. And the other baseline is the Grover GNN transformer baseline, which is the first part of our model. And we find that combining them as well as mutagenicity pre-training achieves a better performing model. And in addition, we noticed that GNN transformer, it's quite variable as measured by bootstrapping, which makes us think that it's performing well in a subset of the data. And then these are our results for CCRAS, our external validation test set. And in addition to the previous model, you notice that here we evaluate against Carcinopred EL. And this is another work that models carcinogenicity. And it was trained on CPDB. And then we find that Concerto outperforms the other models, as well as the baselines. And we think that especially important, Concerto performs well in the low false positive rate regime, which is especially important for prediction modes where you're really interested in maintaining a low false discovery rate, as you would in carcinogenicity. So finally, I'm going to talk about counterfactuals, and we'd like to understand what the model is interpreting as important. And for that, we would want to find a sample that is close in the input space, but far away in the prediction space to our sample of interest. And we use XMOL, that the authors of which are Willowat et al. From 2021. And to generate these molecules, we consider Tanimoto scores as a way of measuring molecular distances. So to find a molecule that is close to a molecule of interest, we have to get some sort of a metric to understand how far away it is. So for that, we use the fingerprint embedding of two molecules, and we take the dot product in the numerator and the sums of the two vectors in the denominator and subtract it, the dot product. And because it's a distance, it's not a similarity, we take 1 minus this. And this is an example of a counterfactual plot. So every single point on this plot is a molecule. And on the x-axis is the Tanimoto score distance that I described earlier. So for identical molecules, you would have a Tanimoto score distance of 0. And then for maximally different molecules, it would be 1. So, and on the y-axis is the carcinogenicity percentile of the test set. And as I mentioned earlier, we want to find molecules that are really close from each other. So they're very similar, but they're very high in differences in carcinogenicity prediction. So here we see that our original molecule is in the 50th percentile of carcinogenicity. And we're interested in identifying molecules that are similar, but either are extremely carcinogenic or non-carcinogenic. And so here we have the base molecule, and in blue, this is the point that corresponds to this. This is the structure that corresponds to this molecule right here. And by observing the differences between the two molecules, we see that in the base molecules, there's the iodine, which is replaced by hydroxyl group, which indicates that our model thinks that if you replace this molecule will become less carcinogenic. And then similarly, this is the positive counterfactual example, which is carcinogenic, which is in the 70th carcinogenicity percentile. And if you replace this hydroxyl group with nitrogen and oxygen, it thinks that it's going to be more carcinogenic. So at a high level, we think that, first of all, molecular carcinogenicity prediction is an important problem. And it is important to think about compound dosage while assessing model performance instead of just a binary classification. And we hope that this counterfactual method for model interpretability can help domain experts interpret the model predictions, what it thinks is important. And then in terms of broader conclusions, so in data constrained settings, as so many life sciences problems are, we found that large models trained with self-supervised learning, so transfer learning from these could be helpful. Pre-training on related experiments, if they're available, could be helpful as well. And then if there's a way to encode domain expert knowledge into the model, that's quite effective. But still the gold standard is if there's a way to get more data, that would be probably the best in terms of model performance improvement. So thank you very much, and I'll take any questions that people have.
Excellent. Firstly, thanks for being on time and also brilliant talk. So while we're waiting for questions, I'll just ask a few. So it wasn't clear to me exactly how you are modeling dosage in your input, or is that part of the yeah, I wasn't sure. I could, you were concatenating a few features together, right? One is from the GNN and the others from
the fingerprints.
The fingerprints. And so where does can you predict carcinogenicity as a function of dosage, or is the dosage implied in the model?
Yeah, so the dosage is indicated by the labels. So here, in this case, the authors of Carcinogenicity Potency Database, they went and did the hard work of assembling primary literature and calculating what the number of tumors is, what the dosage of the original experiment is, and what is the weight of the animal, and using that to have a systematic evaluation of the label. So to have it be a continuous representation instead of like some other databases where you just have a 0, 1, carcinogenic, non-carcinogenic. Is there tumor growth or not?
I see. Okay, I see. I was thinking of whether you are modeling the dosage of the carcinogen itself, but I guess this is more like an effect size of carcinogenicity. Is that correct?
Yeah, exactly. Okay. Yeah.
Great, awesome. And you also mentioned, okay, hold on. There's a question. Okay, so one of the attendees says, what is the rationale for training the model on a continuous value data set as a prediction task and then testing the model on binarized data as a classification task? Did you have to modify the model to output a binarized label?
No, you don't have to. You can still produce a single output, and it can still act as a classification problem because you can just threshold your single prediction. I mean, the rationale is like if we had continuous data, I think we'd much prefer to evaluate on that. But the hard work here, I guess, is actually computing this TD50 value, which requires working with the original data and reviewing, hundreds of papers.
Excellent. I also really like the counterfactual explanation method. Are there have you tried to look for like any global patterns? So I know you have just a relatively small data set, but like if you aggregated these kinds of patterns or multiple molecules, are you seeing some kind of like, is there some continuous pattern that matters, or is it just like, all over the place in terms of like what really drives carcinogenicity up or down?
Yeah, for sure. That's a really interesting question. It's on our to-do list. So I guess the carcinogenicity, people who study carcinogenicity, it's not fully understood what sort of functional groups cause carcinogenicity. But there was a paper that suggested through empirical findings 20 subgroups in molecules that increase carcinogenicity. And we were thinking about doing this experiment, I guess, when we take the top percentile of our carcinogenicity data set and generating negative counterfactuals for them. And then, I guess, assessing in what percentage of the original molecules are these subgroups present, and then in what percentage of the negative counterfactuals do they get rid of these subgroups. And this could be sort of like an independent check on if the model is learning something relevant about carcinogenicity. So yeah, that's definitely something that we're thinking about.
Great. Thanks so much. That was a great talk.
Thanks.
So if you can unshare your screen,
right.
Move on to the next speaker. Excellent. So the next speaker is Joshua Pan from the Broad Institute, and we're talking about sparse dictionary learning recovers pleiotropy from human cell fitness screens.
Thank you, Anshul. Can you see my slides and mouse and hear me?
Yeah, that's great.
Awesome. Well, thanks for the introduction and the invitation to speak today from the organizers. This work was jointly supervised by Marinka Zitnik at Harvard Medical School, James McFarland at the Broad Institute, and William Hahn at Dana-Farber. The key idea of today's talk is that words and genes share a correspondence, which is that their meanings arise from their context. For example, apple is a polysemic word whose particular meaning is resolved via sentence context. Similarly, H2AFX is a pleiotropic gene whose particular biological function is resolved via cell context. So to be more specific, if a cell is treated with a genotoxin, it'll activate H2AFX, but different pathways can result depending on the input toxin. While unsupervised learning of word polysemy is common, unsupervised learning of gene pleiotropy is relatively understudied. So undergraduate ML courses will teach that corpus of sentence contexts can induce word embeddings that obey linear semantics. And so can we accomplish something similarly with genetic data? And the goal of today's talk is to address this problem, both the data types and the potential approaches that'd be useful for learning these kinds of gene functional semantics from data. The data type that we propose using are gene perturbation effect measurements. And this style of experiment involves taking, let's say, a cell line, infecting with CRISPR guide RNAs, and inducing individual gene knockouts in individual cells, and then measuring the outcome of that in terms of some phenotypic measure, for instance, cell fitness. So genes that are critical for fitness will drop out from these screens and get a negative score in these kinds of fitness effect measurements over cell contexts. And it's now very common to repeat these style of experiments over diverse cancer cell lines, for example, or in the presence of diverse chemical insults, starting with a parental cell line. And this is analogous to building that corpus of contexts from which we can infer gene function. And because this is a really causal measurement in terms of the outcome of that gene on that cell function, we think this is a good starting point. And we considered other data types like transcriptomics or PPIs, but they tend to be a few degrees removed from that causal effect on cell function. So how about the approach? So we probably want some low dimensional vector embedding in which genes are vectors in some latent space. In 2011, Günter Wagner and Jiangji Zhang wrote a very influential review that outlined a few criteria for inferring pleiotropy from these high dimensional biological phenotypes. So their first criteria is that the representation should be sparse. So evolutionarily, genes tend to have a few functions that are constrained and that are context specific, but not too many. The second is that any latent variables learned from the high dimensional data ought to be biologically meaningful. So if we do something like PCA and we get some really nice numerical factors, if those don't align to biology, then we're not counting phenotypes correctly. And third, we need to account for redundancy. So if we screen 100 cell lines, we're probably not going to get 100 independent cell phenotypes. So there are correlations between cell contexts that need to be taken into account. So inspired by these criteria, we looked to a method called dictionary learning, and we developed an approach that we call Webster. So Webster takes as input a matrix of fitness data. So every row is a cell context, every column is a gene, and every entry represents the essentiality of that gene in that context. So the output of this method are two low-rank matrices. The first is this dictionary matrix here, where instead of having genes as inputs here, we now have derived some latent biological functions automatically from the data. And the second matrix is a loadings matrix. So the task is that every gene effect present in the input data needs to be reconstructed in terms of a mixture of up to T of the elements in the dictionary. And T is a hyperparameter that you can set. And so this is equivalent to an L0 penalty on our representation matrix here. So there are a few other augmentations that we added to this task that allow us to preserve interpretable relationships between genes. So Anshul's lab and others, hopefully on the call, have studied this. And gene effects have correlations that are highly informative of their biological functions and reflect sort of the modular structure of these kinds of pathways that are represented across cell contexts. So these columns naturally form a coessential graph or a co-fitness graph that has some structure to it. So what we want to do is we want to be aware of that graph as we're learning these gene to function relationships. And so two genes here, let's say in green, that are similar in the input data ought to map to similar functions in terms of their coefficients in the low dimensional model. We can do a similar trick to manage cell context redundancies. So there are correlations across rows that are informative of similarities between biological contexts that are potentially redundant. And so if we, for example, have two cell contexts that are highly similar in the input data, we want to preserve that similarity in our low dimensional matrix here, where we have functions instead of genes here. So to put it all together from top to bottom, our Webster approach is as follows. So there's some standardization and pre-processing involved. You want to filter out genes that don't have a phenotype. But again, the input is this pre-processed fitness data here, this matrix Y. The outputs are these two matrices that I've described before, the key hyperparameters being K, which is the size of our dictionary, the number of latent elements to look for, and T, which is our degree of sparsity in our gene reconstructions. The objective function, the first is standard, sort of a reconstruction error in our low dimensional representations of that input matrix. And then we have our two graph priors as augmented objectives in our objective function. So again, this is a graph prior over the columns of X induced from the correlation structure of columns in Y. And this is a graph prior over the rows of D induced from the correlation structure of the rows in the input data Y. So let's go through some applications on real world data. So Olivieri et al. Performed a set of fitness screens in the presence of genotoxins. So they started with a parental cell line, RPE1, and they treated with different DNA damaging agents. So for example, UV light will cause DNA damage in cells. In order to repair that damage, certain pathways need to be activated and present. So by knocking out genome-wide in these treatments, they were able to identify which pathways were critical for repairing the damage induced by treatments. So in this matrix, treatments are rows, genes are columns, and each entry represents the essentiality of that gene in the presence of a treatment. So once again, our input data is this matrix here, and the treatment names and gene names are elided because they're not necessary for the factorization. And after a hyperparameter sweep across K and T, we decided to recover a dictionary of size 10 and use 2 dictionary elements each to reconstruct each gene effect. And so this is our approximation of that matrix. You can see that we've captured some broad patterns in the data. So this is what our dictionary matrix looks like. As advertised, there are 10 columns, 10 latent variables that were automatically learned from the data that we hope align to biological functions. And again, each function has been inferred to have a fitness effect across a certain profile of treatments in the input data. This is our gene to function loadings matrix. You can see it is quite sparse. Every column has only 2 non-zero coefficients. And this is the way that our model reconstructs gene effects from the input data in terms of these dictionary elements. Now, this gets interesting when you start to add literature annotations that were hidden during model training. So for example, there have been some genes annotated as nucleotide excision repair genes, classically defined in the literature. And we can see that in our first latent variable here, we've captured a cluster of genes that seems to map to this particular biological pathway. So that is the inspiration for our name for that latent variable here. And if we look across our dictionary, we can see that UV induces a fitness effect in the absence of nucleotide excision repair, function number 1. And that is indeed the case biologically. UV causes DNA adducts that are removed by the excision. So what does this model have to say about individual gene effects? So here's an example. So H2AFX is a DNA damage protein, and its treatment, its fitness effect over treatments has been measured here. So our model approximates that profile as a linear combination of 2 out of the 10 dictionary elements. The first of which captures the effect of homologous recombination in the data, and the second corresponding to the effect of end joining. And both of these were again learned automatically from the structure of the data itself. And so these are some of the treatments that seem to induce the necessity for that pathway the most. Well, if you remember from the intro, this is that pleiotropic gene I introduced. So H2AFX can activate an ensemble of functions depending on the cell state. And so again, this relationship was hidden during model training. The final notable thing is that this latent space now allows us to perform this kind of vector arithmetic I was talking about. So if we take the treatment profile of fitness effects for H2AFX, subtract a dictionary element corresponding to end joining, which is a pathway that we inferred, add a dictionary element corresponding to Fanconi anemia, we get some other value. And that value actually looks a lot like the gene RAD51B, which is known to be part of this pathway, as well as homologous recombination, which it shares with H2AFX. And so we can see the correlation between that synthetic profile and RAD51B measured profile here. Okay, so I'll briefly touch on 2 other applications. The first is that we scaled our approach to a much larger data set called dependency map. Instead of treatments over the same cell line, dependency map is a set of screens over diverse cancer cell lines, each of which have their own unique fitness adaptations and different combination of activated pathways. So it's a great testing ground for this method. And again, they've treated it with a CRISPR-Cas9 guide library to induce these gene effects. So we recover a dictionary of size 220, which was a hyperparameter that we optimized on this data set. And then we used for each gene effect up to 4 of these dictionary elements for its approximation. And so once again, we can recover these sort of interpretable linear semantics. So SHOC2 is a signaling protein downstream of multiple upstream activators such as KRAS and NRAS and EGFR and FGFR. So this is the measured fitness effect of knocking out SHOC2 across 675 cancers. And so our model approximates that profile as a mixture of 4 latent variables stored in the dictionary, each of which captures the biological effect of one of those 4 pathways. So you can also do some exploration using embeddings and UMAP for visualization. So each of the gene effects that was measured in this data can form a data point in this low dimensional embedding here. And then each of our inferred functions in the dictionary has an inferred fitness profile. So those can also sit in the same space. And so here's that approach. So this is a UMAP embedding of the top 3,000 or so fitness genes in the data that was used as the input to Webster. And you can see that they're sort of non-uniformly distributed across this embedding. And if you add on the functions that we've learned automatically, you can see that they kind of form like cluster centroids. And this is exactly what's happening in high dimensions. A group of genes that are collinear will become maps to a learned representation of that module, which is what we call a function here. And this becomes really fun when you start to add our human annotated labels for these. So for example, there's a group of functions down here that correspond to unique processes in the mitochondria that are important for metabolic regulations in cancers. So as our final application, just a brief vignette on using compound data. So as we've learned throughout many talks today, when we discover a compound that's interesting, we'd like to know what its potential biological mechanism is. And so one common approach is to treat cancer cell lines with this compound and see which cancer cell lines survive and which ones don't. Now those profiles tend to be noisy. So it's possible that we could use the functions that we've derived from our very specific and very clean CRISPR knockout data as kind of a reference latent space with which to model these potentially noisy compound treatments. And so in order to try this, I used PRISM, which is a data set in which compounds were treated over cancer cell lines and their sensitivity was measured. And then I took our dictionary from our CRISPR analysis and I kind of fixed it. So then the only variables here were these coefficients. Similarly to our genes, we try to learn a representation of our compound sensitivity in terms of these dictionary elements. And so as a result of doing that, we can now align compounds into our embedding space. So now you can see there's a third layer in sort of our visualization here. And for instance, from CRISPR data, we automatically learned a representation of BRAF signaling. That's one of the 200 elements in our dictionary. And we can find that both gene effects and compound sensitivity effects are both collinear with that function latent variable. So they kind of hit the same cancer cell lines and maybe represent the same pathway. And this is true for multiple other compound classes. So to conclude, we think unsupervised learning of gene pleiotropy is tractable. We recommend using high D gene perturbation measurements as a starting point for such tasks. And for the embedding approach, consider using sparse approximation for its interpretable linear semantics that you can get from the data. And so I'd like to thank once again my mentorship team and open up for any questions. Thank you for your time.
Thanks, Joshua. That really spectacular talk. I mean, great science, but also absolutely really wonderful presentation. Thanks for that. So while we wait for questions, I'll just ask a few. So I was wondering, so I'm sure you did the counter experiment where you didn't regularize the model based on the two graphs, right? The cell-cell graph and gene graph. And do you still see it pick up at least partially relevant modules, or are they kind of all over the place? I'm just wondering, how strong the contrast is. I love the idea of semi-supervision of sorts, but I'm just curious, like how free is the space if you don't constrain it?
Absolutely. So just qualitatively on the smaller data sets, like the genotoxic fitness screen, where there's only 30 conditions, the non-graph regularized version seems to work really well. In the very large cancer dependency map, just like your work showed in Nature Genetics, those modules can be very tight in a very large space of like thousands of genes. So that's when the graph regularization really kicks in, because it helps us find our latent variables and align them to biological modules. The last side note is that whenever you do this graph regularization, you have a less variance in your model output. So it tends to find more reproducible latents, but at a computational cost.
That's really great. Let's see, there's a question from Alan. What is the motivation for using a sparse representation versus letting the model set low values for particular entries of the matrix?
Sure. So I'm trying to interpret that question. I think you're asking maybe why L0 versus L1 or L2, which are different ways to induce sparsity. So yeah, that was a design choice in the model, if that's what you're asking about. And I just found that the interpretability was higher when we imposed like a hard sparsity constraint. So if we set T to be equal to 1, that's equivalent to hard clustering. And when we set T to be equal to 2, that relaxes those assumptions. So you can think of Webster as like a soft clustering algorithm. The L1 and L2 and elastic net penalties didn't do enough to drive interpretability in my hands.
Excellent. Last question. Bruce asks, do you have a sense of the strength of classifying inhibitory versus cytotoxic effects? And have you gotten into high additivity combinatorial agent synergy?
Excellent question, Bruce. And you must have seen my hidden slides. Indeed, the compound dose is very important for this projection. So if your compound is treating at a very high dose, that looks like a cytotoxic effect that kills a lot of non-specific cancer cells. And then all of a sudden that compound alignment fails to work. At a low dose, the opposite happens where there's just no effect. So I showed you kind of these optimized dose profiles, but in my supplemental tables and on the paper online, you can find kind of dose curves where that projection score varies in terms of compound dose. And no, I have not looked at compound synergy.
Excellent. So in the interest of time, we'll move on. Thanks again for that really nice presentation and work. The next talk, the last talk for this session, is from Ran Zhong. I hope I got the name right. It is on semi-supervised single-cell cross-modality translation using polar bear.
Okay, thanks for the introduction. Can you see my slides?
Yes, but it's not in full screen mode right now.
Okay.
Okay, there we go. Yeah, perfect.
Okay, thanks. Hi everyone. I'm Ran Zhang. I'm a postdoc in the Novo Lab in the University of Washington. And I'm going to discuss about our current work on semi-supervised single-cell cross-modality translation using polar bear. This is a collaboration work with Ticia and John Flip in the Google Research Brain team and my advisor, Bill Noble. So single-cell sequencing-based omics spans multiple modalities. Each data assays or data modalities here captures a unique aspect of single-cell regulation. However, in most single-cell assays, only one type of activity can be captured for a specific cell. In order to gain a more complete knowledge of a particular cell, we ask if we can infer the missing cell profile in the missing modality based on the measurements from the known modality. And this idea is made feasible by the emergence of single-cell co-assay data in the recent years. Here, based on the preliminary survey of the co-assay data in the previous years, where for a specific cell, it has more than one modality mirrored. So we decided to focus on the co-assay in between gene expression and DNA accessibility, since this type of co-assay data are currently most prevalent. So here, our goal is to learn a translator in between the co-assay data so that we can, with cells that are only mirrored in one data modality, we can use this translator to predict the missing profiles. And in practice, however, single-cell data is quite sparse. And the co-assay data we can get now are quite challenging to produce and lower throughput than the single assay data. So we hypothesize here that a semi-supervised framework leveraging both co-assay and single assay data will facilitate the cross-modality translation. And with that idea, we propose this polar bear method, which is a semi-supervised framework that can leverage this unlabeled single assay data into the training process. So here, in the first step, we will train an autoencoder for each data modality, including all the single assay data, to learn a more robust representation of the single cells. And then we can learn a translator in between the embedding layers of the autoencoders in a supervised fashion using the co-assay data. That said, we are not the only one that is working on this field. So here is a brief summary of the current methods that we are aware of that performs cross-modality translation. However, most of these existing methods are trained only using the co-assay data. We do notice that this multi-VI paper, which is published, which is released in bioRxiv recently, does allow input as single assay data. But it is not demonstrated if adding the single assay data from unrelated studies would augment cross-modality translation. And with that, let's go to the detailed workflow of polar bear. So we first collect a bunch of single-cell assays, single-cell RNA-seq and single-cell ATAC-seq profiles, including both cells in the co-assay and cells generated by the single assay data. And then for each data domain, we use all the cells to learn a variational autoencoder, where so these architectures are taken, adapted from the ICVI and PIC-VI methods, where we specifically model this batch factor and sequencing depth here, so that the embedding layers we learned are not biased towards those terms. And for RNA-seq, we input the raw counts and estimate it by the zero inflated negative binomial distribution. And for ATAC-seq, we input them as binarized profile and estimate them by the Bernoulli distributions. So after we pre-train the model on all the data, we can learn a single neural network, a single translation layer in between these embeddings from the two data modalities based on the supervision of the one-to-one cell correspondence in the co-assay data. And once we train the polar bear model, we can apply it in the scenario where we can predict the missing domain profiles. Based on the existing single assay profiles, we can couple the encoder of this known modality with the decoder of the missing modality together with the translation layer we learned. So that we could make predictions on the cellular profiles in the missing domain. And with that, we can generate interesting hypotheses, for example, cell level differences or group level signatures for the genes and peaks. And besides, we can also use polar bear to align single cells across data modalities. So here we can project the single assay data in each data domain to a shared embedding space in the polar bear architecture, and then we can map the cells across these modalities. So, so next I'm going to go through the details of these applications and the evaluations. And here we specifically focus on the snare-seq co-assay data, which has around 10,000 cells captured by the RNA-seq and ATAC-seq in the adult mouse brain. So we also collected several large-scale single assay data sets in the same tissue. And here you can see that the data sets, the data quantity we can get for the single assay are way larger than what we can get for the co-assay data. And to evaluate, we tested in two scenarios. In the first scenario, we hold out a random 20% of cells in the snare-seq as test set. And in the second scenario, we test if this model can generalize to predict on a unseen cell type. So we hold out an entire cell cluster from the training process. And to evaluate, we first try to evaluate if we can recapitulate the single cell RNA-seq prediction based on single cell ATAC-seq profiles on these random 20% holdout cells. These cells are not used in the training process. So, so instead of calculating an overall Pearson correlation score between the predicted and the original profile, which is what most previous methods does, we argue that in that way, the evaluation metric can be highly driven by the overall gene expression pattern, the average gene expression across all the cells. So here we really want to tease out the individual cell level differences. So for each gene, we calculate the Pearson correlation of its predicted profile and the true profile across all the cells. And we compare our polar bear's prediction with another state-of-art method, Babel, which is published in PNAS this year. So here each dot is represents a gene. And here we can see that in the majority of the genes have improved Pearson correlation when they use the polar bear model. And next, we also want to directly address the benefit of our semi-supervised framework training with all these single assay data collected from public database. So we compare polar bear with a variant of polar bear, which we named the polar bear co-assay, which is trained using the same architecture, but only on the co-assay data, as what is previously did in other methods. So here we can see a very clear improvement when we leverage the single assay data and the semi-supervised framework during the training process. And again, we can evaluate in the opposite direction, where we try to see if we can recapitulate the single cell ATAC-seq profile based on the known single cell RNA-seq profile in the test set. So here for each gene, we calculate the AUROC of its predicted profile based on the true profile across all the cells. And again, we observe that polar bear significantly outperforms Babel and polar bear co-assay at recapitulating the peak level differences, the peak-wise differences. So, then we test in the other test scenario where we hold out an entire cell cluster from the training and make predictions on this cell type, whose co-assay is not used in the training process. This is trying to see if polar bear can generalize to the cases where we predict on a new cell type that is not seen before. So, and we specifically define this unseen cell type the same, using the same way Babel did, so that we could make a fair comparison with them. And again, we calculate the gene-wise correlation as well as the peak-wise AUROC when they predict on each direction. And we observe similar trend that polar bear significantly outperforms other methods in terms of recapitulating the individual cell level difference within this unseen cell type. So in this scenario, besides caring about the individual cell level differences, we also want to see if we can recapitulate this gene expression signature for this entire cell type. This could be the marker genes or the cell type specific genes, and we want to make sure that our prediction can recapitulate this. So here we define the cell signatures as a significantly highly expressed genes in the unseen cell type. And the true labels are generated based on the differential gene expression pattern in the true profile. And here we use the different models to predict the profile and generate and predict differential gene expression pattern and evaluate this predicted differential expression pattern on the true differential expressed genes that we get based on the true profile. And here, based on the precision recall curve, we can see that polar bear is the best at recapitulating the gene signatures of the unseen cell type. And finally, we also found that as an added benefit of this translation model, we can also use polar bear to align cells across different modalities. So here we start from these single assay profiles from the test set, and then we project them to the same embedding layer in the polar bear architecture. And here is a UMAP visualization of the cells in the same projected space. And then we can ask if the corresponding profiles for the same cell can be mapped closely together in the shared space. So here, a shorter line indicates that the corresponding profiles for the same cell can be projected quite closely into the shared space. Meanwhile, a longer line indicates that the alignment is not as ideal. So we further quantify this alignment based on the FOICTT-M score, which represents a fraction of samples closer than the true match. So here, we can start from a specific cell query in one data domain and ask how many cells in the other domain are mapped closer to the cell compared to its true match. And the lower FOICTT-M score indicates a better match. So here, the one-to-one correspondence is not used to force the mapping, but only used to evaluate the alignment performance. So here we sort the cells based on their FOICTT-M score in each data modality, and we observe that in general, polar bear performs better at aligning these cells across modalities. And we also test this in the unseen cell type scenario, where we really try to map the individual cells within a relatively homogeneous cell type. So here we can see a more obvious improvement using polar bear, the semi-supervised framework, compared to using the models that is only trained on the co-assay data. And to conclude, polar bear semi-supervised framework improves cross-modality translation performance by leveraging the tons of single assay data that we could easily get from the public database. And also polar bear can match cells across modalities. And we also demonstrated that it can recapitulate the individual cell level differences. For the future work, we can generalize this model to various datasets, biological contexts, and data modalities. And also, given there are a large compendium of public single assay data, we can adapt this model to align those available single assay data out there so that we could map them and generate interesting new hypotheses, for example, gene peak correlation. And finally, I think it would be nice to apply polar bear's framework to make predictions on some disease models with the hope that we could gain a more complete insight of disease etiology. And with that, I'd like to thank our collaborators, Leticia, John Fleet, and in the Google Research, and my advisor, Bill Noble, and also members in the Noble Lab for their valuable suggestions, especially to Yang, Anu, and Dijun and Aisha for their valuable help. And this manuscript has been recently released to bioRxiv. And with that, I'd like to take any questions.
Excellent presentation, Ron, and great work again. So you got quite a few questions. I'll just try to read them out one by one. So Alina asks, are there any challenges in using single assay and co-assay data together as one input to an autoencoder? For example, is there any specific pre-processing that's needed?
So that's a great question. So we didn't pre-process the data since our method allows the input as raw counts. But in the case of integrating them, I think one challenge is about the batch effects, the potential batch effects. So in our current model, we actually, we specifically account for the batch effects so that, and we can see that the batch effects are kind of like a somewhat removed, but there may be more effort needed to better correct the batch effects by forcing them to project to the same space if we, in some scenarios.
Excellent. The next question is, how does polar bear compare with other existing methods in addition to Babel? I guess you showed this in the talk. But...
Yeah, that's a great question. So actually, there are several existing methods. We chose to compare with Babel because Babel directly address the single cell RNA-seq and the tag-seq translation task, and also it is directly applied to this snare-seq data set. So we think it would be a more fair comparison. For other methods, we did try multi-VI, but we couldn't, since the feature space in this data set is quite large, so multi-VI kind of runs out of memory. And so we decided that instead of comparing with all the current methods, the main point of our current work is that adding the single assays from the public database can really augment this translation. So we think this idea could be adapted to other current methods to help improve their performance. So yeah.
Excellent. Without adding single assay data, does Polar Bear still outperform Babel?
Yeah, that's a great question. So we actually in our we did multiple evaluations, and what we observe is that Polar Bear in most of the times outperform the Polar Bear co-assay, which is trained only on the co-assay data. In most of the times outperforms Babel, but there are scenarios where there's no significant difference.
Thank you. David Kelly asks, can you interpret the cross-modality translation? For example, are attack sites near a gene used to predict its RNA?
That's a great question. So currently we don't we don't kind of assume anything. So yeah, that would be interesting to check if the close peaks contributes more.
Gerald Pau asks, did you look if any of the latent factor variables of the variational autoencoder bottleneck that you find correspond to something interesting that can give you biological insight?
That's a great question. We haven't looked, but that's something really interesting. So we will check.
Bruce, I don't know, asks, besides better accuracy of cell classification, do you see evidence for functionally significant subclasses identified by multimodality profiling?
So I think so currently we did try something at for example classifying the cell types, which is kind of I think for single cells the function may be a little hard to interpret. So we kind of so we did try to use this method to apply to the cell type classification. For example, based on the single cell taxic profile, if we can predict the cell type marker genes to be highly expressed in the corresponding cells, and we do find Polar Bear outperforms other methods in this case. So this is kind of going to the way of the functional, but not exactly yet.
Excellent. I think that's the last question we had. I'll just add one more similar to what David asked. So I guess I was thinking more in terms of in the training itself, is there any way in which you think it would be useful to add constraints between the RNA features and the attack features? So I think David's question was can you take your current model and then interpret it to identify interesting correspondence between features? I'm actually asking whether you can in fact have some kind of graph constraint in the model itself, where you kind of have genes nearby peaks and peaks nearby genes have some relationship, and see if your model can do even better, because you might get interpretability and stability, and maybe better performance that way too. Or do you think that's not necessary?
Yeah, I think that's a great idea. I think we are also I think it's interesting to kind of incorporate some prior knowledge, for example, the gene peak the prior knowledge on their relationships, and also maybe sequence or other prior knowledge to help guide the translation. Yeah, that's something interesting in the future.
Great. Fabulous. Thanks so much again for that really nice talk and great work.
Thanks.
And thanks to all the speakers. I think all the speakers today have been absolutely fantastic. So I just do want to again commend you for your efforts and great presentations. So I think that's the end of our day today. But please don't go away permanently. There is another day tomorrow, and we're going to have a very exciting industry panel starting off right at 9 a.m. Pacific time, followed by spotlights, a poster session, another keynote, another set of orals. So please do join us tomorrow. Sarah, did you want to say anything before we head out?
No, I think we're good. So see everybody tomorrow at 9 a.m.
Okay. See you all soon. Bye.